Recording is on. So welcome to the meeting of the Open Source Drug Discovery for Malaria project on Monday, May the 13th at 4 p.m. Sydney time. Just for those of you who are present who want the briefing, this meeting is being recorded and streamed. Anybody can take part, but a copy of it will be loaded up onto YouTube once we're done. So just please remember that it's live, and if you so please be careful saying anything that is either unwise or commercially sensitive or anything else, because we're live now. So this is a regular project meeting, and it's been about 2.5 months since our last one. In that time, there's been various things happen. So lots of compound synthesis, biological evaluation, and it's time to take stock of where we are. Unless anyone's got anything they would like to say at the start, then we can get stuck into the agenda, which is in the discussion note window on the screen there with the appropriate links. And the first thing that we need to do is just check on the action items arising from the last meeting on February 26th. And the link is there. At the bottom of the page is the to-do list from that meeting. And unless anyone had any questions about this, I was just going to go straight through those quite quickly, just to sort of update people on the progress, and we can put a written version of the updated to-do list on the minutes for this meeting, as is usual. So on that list, which hopefully everyone's got there, the link, as I said, is on the discussion notes window. The first action item was to have this aryl sulfonamide biologically evaluated. So yes, excitingly, Patrick Thompson did finish making that aryl sulfonamide. And a couple of other analogs. And we organized via Paul to get that evaluated in Dundee. And the data came back from that saying that, perhaps not surprisingly, for those of us who've been in this, been dealing with the aryl perols for quite a while, that the change from the acid to the sulfonamide eliminated activity. So the sulfonamide and the two analogs were inactive, completely inactive. Now, that's as far as I know, that is the last bit of biological evaluation we were waiting for the completion of that series, essentially. So that's the last analog that we were planning on making for that set of compounds, but it's inactive. So it adds to the data, which says that it's extremely difficult to replace that ester with another functional group. I don't propose to go into too much more detail on that point, other than to say that that will, of course, be included in the paper that is being written on the first series. Oh, yeah, Murray, you were talking about the sulfonate. I've enabled your audio, Murray, if you wanted just to address that point. I know you're a fan of this compound. If you wanted to say something about that. If not, then just say, but otherwise you can talk about it. Enable your audio, Murray. I think maybe you have to enable it yourself. Up at the top of the screen.
Hello. Yeah. Sorry, my computer just crashed. Froze there. Yeah, the sulfonate just appeals to me because we've shown from quite a lot of the other compounds we made, when we have a nitrogen in that position next to the carbonyl, activity drops off, whereas the ester. I think sulfonate, and I think Matin is in the process of making it, so I think the sulfonate is a nice compound to make. And if Matin is doing it, then it's not too far away, and it's not too hard to make. Last time I spoke to Matin, he seemed to think it was just as, well, not as easy, but the same level as making the sulfonamides that Patrick's made.
Okay. Of course, we are still, writing this paper, so we're not completely done yet. And of course, yes, Mathen is finishing up his grad dip here, so he's kind of addressing that compound anyway. So certainly, as Alice writes in the window there, we can certainly wait to see where we get to on that. Unless anyone's got any real complaints, we do have a little bit of time while we're finishing off writing. We can pursue it to sort of close the cycle.
So. Sounds good.
Okay. The next item on the to-do list was reduce OSMS 109 to the aim and evaluate potency. Murray, that was done, right?
Untested, I can't remember. It wasn't that fantastic, was it? And
yeah, it had some activity. Had some activity. It wasn't as bad as some of the others. It had some,
yeah,
but it wasn't.
the synthesis. Sorry, I was fairly confident that that was um, good for testing. That's why I sent it off. Although I almost to have it fully characterized, it needs to be synthesized in HPLC or something, because the final purification was a bit of a mess with solubility and everything. But I was happy with the NMRs to have it tested.
Okay, but if we're going to include the biological data in the paper, we're going to have the data.
It's easy enough. It's just one step, and there's plenty of the nitro compound, so I've just run out of time. But that's easy enough to do.
Okay. Okay. All right. Number 3 was for the methoxy substitute in the enable compound 111. What we were going to do here, we kind of stalled on the idea of having this biologically evaluated in vivo because we wanted to see what the solubility and metabolic data were like. And that still sits with me, so I still haven't done that. I need to make sure that I need to contact Stu Charman to ask about solubility and metabolic evaluation before we think about committing something like that to in vivo analysis. So that's Paul Willis and I corresponded a little bit about that, and the action items on me for that. Number 4 is summary for the reasons for the stops on the aryl-pyrrole series. And the work to be continued on the experimental section of the paper. I think both of those at the moment are on Alice as to-do items. I think she's still plugging away at that, making large amounts of progress in the paper, and we still haven't got a good summary on the reason for the stops, which we will get soon. Number 5 was check on the need to resynthesize 106. So that's the Jimmy compound, right? The initial aminothiopyrimidine compound, I think. And again, I haven't approached Sue yet about that, but we have that in stock, I understand. So Murray and Althea, I think, have created a stock of that now. Is that right? Murray, your audio is still on if you want to talk.
Althea's just got 70 megs, yeah.
Okay. 70 megs, so we have plenty for what we need to do for that for the short term. Okay, so the action's on me to get that evaluated by Sue again. Number 6 was a short list of thionopyrimidine compounds, request input from the community. We kind of did that, although it was a short consultation, and that's the subject of the next action item, number 3, which is the aminothiopyrimidine series. We did get a set of those evaluated, so I guess that was done. The number 7 was a summary of Suzuki reactions trials to date on the thionopyrimidine series. That the need for that was eliminated when the microwave chemistry started working for Althea. So to some extent, that was made unnecessary because we solved the problem, which is why that didn't appear. We began to assemble a table, but it didn't need to be sent out to people because it started to work. The sledgehammer of the microwave, I guess it always works. Number 8 was post-procedures for the synthesis of the chlorothiopyrimidine. I guess that's also been put on hold because we want to check which compounds we're proposing for the next round. So if that key compound, the chlorinated thionopyrimidine core, looks to be useful, we can go back to the CRO sector, but it is commercially available for a reasonable price. So I think we'll put that on hold for the moment and maybe revisit it if it looks like we're going to need large amounts of that material. The approach, though, of exploring with the CRO sector the synthesis of required starting materials is still a good one. We just want to make sure that we deploy it at the right moment and for the right material. So I think we pause on that for the moment, but we may come back to that. 9 was explore the use of NIME with project data. Murray's been active in doing that and has been bringing Alice up to speed on what NIME can do, which is really great. So I guess that's done. 10 was to obtain a quote for the development of the project landing page. I'm in the middle of discussing with web developers about getting a project landing page done. So that's still with me, but that's been started. 11, explore whether GitHub could be used as a project coordination. Well, we've been doing that in the last few days over the weekend, using GitHub as a project management tool. So Paul, we've just to bring you up to speed, since about Thursday or so, we've been using something called GitHub for seeing if that could be a way of having a website that deals with project management. That is to say, all the to-do lists is an active set of objects, and managing the project more closely, so we can say when action item has been achieved, and we can group things together under milestones, rather than having a text-based to-do list. And we've only been doing it for a few days. The experience for me has been extremely positive, and allows us much better control of the to-do list and who is responsible for each item. Alice and Murray have also been involved in doing this, so I'll ask them their opinions in a moment. We've also had already over the weekend input from somebody else who's monitoring the project, who gave us some input on the GitHub website. So we were nervous because we didn't want to introduce another website where people had to have a login, but on the other hand, it does seem like a step forward in terms of having an active to-do list that people can all see and collaborate on, which doesn't require maintaining a sort of static list on a wiki. Alice and Murray, did either of you want to talk about it? It's early days, but did either of you want to just talk about your experiences of that so far?
Yes, so yeah, we agree with what you said. We don't really want to have another website, another login and everything, but I prefer this method than as you were saying, the list on a wiki, which can go out of date very easily. This setting tasks allows people to write comments and stuff, which is easier to follow. So I personally don't know of any other sort of online project management tool. So this is free and open, so it seems the best option at the moment. But
it's also got a very large user base, right? So it's an extremely widely used tool in software development.
Yeah,
as well supported.
But if someone knows of something better or something that can be incorporated into another website, then maybe, or as we were discussing before, if there's a way you can pool those jobs and have them appearing on websites or Twitter feeds or RS feeds, it'd be nice, which can probably all be done, but that's beyond me.
Sure. Paul, I wanted to ask you, my assumption is that in pharma, when a project is being done, there is a project management tool which helps the team coordinate all the activities, but I've never seen such a thing. Do you have a sense of whether tools like that are widely used and are essential to the quick resolution of a project?
I mean, in my experience, they are used, but they tend to be used more in the development or in the phase where comrades are approaching development, where you have multiple interdependent activities which you need to track. In my experience, most discovery projects within pharma don't use tools. They tend to just use distributed minutes and perhaps a website with them on. But if we think it could be useful in this case, I think it's worth exploring. I guess it is just that concern if it does need another login, whether that will actually act as a deterrent from people using it rather than a benefit.
Sure, so the login is required to contribute material to the website. It's not required to read anything. That was a key thing. So all of the action items and what we're currently looking at is viewable by anybody, but commenting requires a login, and I guess that's unfortunate, but that's the way it is with some of these things. One thing I like about it is the ability to sort of group items together into a kind of milestone. So for example, we have one thing at the moment where a milestone is the completion of the first paper, and anything that's relevant to doing that is then grouped under that as being issues, and anybody can comment, anybody can open and close the issue. It seems to work quite well and have a lot of the functionality I was hoping for. Again, it's nice to sort of reveal where we are up to in various things. One of the suggestions way back for the landing page, in fact, was to have some of the things that we were currently working on with little milestone bars, sort of like these, Murray put it like the church fate kind of raising money kind of bar that goes up and up. that has that kind of built in, so you can see how close you are to finishing milestones. Little things like that. I mean, it's quite a well-developed platform. So, I mean, I propose to continue using that for the moment because I'm finding it very useful, and we can revisit at the next meeting to see how that's gone, to check just to make sure that it's actually helping us with productivity and isn't acting as another drain on the time.
So the plan at this stage would just be to use it within Sydney and then give it another month, and then if you think it's good enough, potentially launch it to the whole project next month?
Well, I mean, it's being used at the moment for everything that we're doing. So anything that is required to be done, we're sort of gradually loading those up onto this website. So it should eventually contain everything. Already some of these things that were action items for this meeting have been closed. So, I mean,
the stage at which you actively publicise it and make that main repository on the website.
Yeah, definitely. No, good. No, that's a good point. So I think that we probably trial it for another couple of weeks maybe, and provided that there's no snags and it doesn't become overwhelming. I mean, if it's positive, then we publicize it more heavily. Yes, it is interesting that what we're doing has been discussed quite a lot in sort of open science forums. We're not the first people to have this idea about using the software GitHub for managing projects. And in fact, there is a team that's adapting it specifically for managing science projects, but it's in some software called SciGit, I think, and it's in beta, and I think, as far as I remember, it's only for PC at the moment. So I think people are already trying to address this, but from what I can tell, the software as it stands, without any sort of tweaking, is already fulfilling a lot of the needs that we have for managing who is doing what. So in that sense, I don't see the need for changing it much. But the test drive has only been active for about 4 or 5 days. So let's see. Let's see how we get to. But yes, if we carry on for another couple of weeks and if it's positive, we'll spread it around more and we'll try and get people involved. Like I said, there already has been someone outside the core team who has contributed on that website, so he's found it and contributed there without any difficulty, without us telling him what to do. So it looks to be quite intuitive. Anyway, we'll see. We'll update next meeting, but if it goes well, we'll start trickling the news feed out trickling the to-do list out to various people and seeing what they think. Okay, so the number 12, we contact previous attendee from Roche. That was something on Alice's to-do list. So that was a previous attendee from Pharma. I'm not sure if Alice got around to doing that, but she will if she hasn't already. And 13 was done. That was just a meeting recording. 14, exploring the involvement of undergraduate communities in the synthesis of compounds. That's happening right now. So there is a team of 49 undergraduates at Lawrence University in the US who are currently making some derivatives of the near-neighbor set of compounds. That is to say, some of the compounds that we thought were most polar and most active from the near-neighbor set of compounds. Now, that's a set that we're no longer targeting actively as part of the research project, but the chemistry involved was extremely straightforward and suitable for kind of the undergraduate class that my contact there was interested in trialing this with. So they are right in the middle of that. They've made a few pyrroles, and they're now, I think, planning on doing the formylation reaction. And so they're right in the middle of that, and he promised me he's going to share some data from the students when they can. So they're not currently posting the data in the open domain, but they are doing the experiments. So what I'm hoping to get from him is some information about where the students are up to. Now, if that works, and if the students do get some compounds out, and if we can then perhaps evaluate them, then we can show that cycle works, which would be pretty exciting. So again, that's in process, and I'll update with a post about what the students are doing as soon as I can. 15, I haven't done yet. We contact Mike Pilastri. I keep meaning to do this, but I won't. If Mike's watching, sorry, I haven't contacted you yet. And 16, new sources of possible synthetic chemistry input on the thionopyrimidine series. We know that Patrick Thompson is interested in transferring over to some of these compounds, but I guess it depends a little bit on what we decide in the next on the agenda item 3 about what we're going to do with this series. So maybe we should come back to that when we discuss that. Okay, that's action item. That's agenda item 1, which is actions arising. Unless anyone's got anything else they want to say about that list or things that we didn't get around to talking about, we can proceed to number 2. which is actually a short item because it discusses the aryl-pyrrole series. And again, I guess we've already briefly talked about this. So the sulfonamide that Patrick made was inactive. We think that the sulfonate might be a compound which would be of some value and which is currently being made by Matin. As far as I know, that is the last of that series, just for the moment, with the caveat that we are still looking at some solubility of a couple of other compounds with SU. But we're not really putting much synthetic chemistry effort into that. We're not putting any synthetic chemistry effort into the aryl-pyrrole or the near-neighbor series at the moment, on the understanding that we're intending to write that stuff up for submission of the paper. So I don't think I've missed anything out there, but that's where we currently stand with that series. If that's okay, then we can move to the main item on the agenda, which is number 3, which is the new data associated with the aminophenylpyrimidine series. So as background here, so there is a link there, and there were 4 picture files which were produced by Murray and Alice, which summarized the biological data that we received back on these compounds, as well as some minor comments on solubility that we received back from Vicky's lab. So hopefully everyone has those sets of data. The data are not complicated, but I guess trying to tease out some information about SAR is complicated and is quite difficult. The background here is that one compound, so an aminophenylpyrimidine with an attached aryl sulfonamide, was in the original GSK Trescantos set, antimalarial data set. TCMDC 135294 was the compound, which was the hit compound, and which was listed as having a potency of 150 nanomolar. We made that compound, that's OSM S106, so that's the compound we briefly mentioned earlier, and tested the activity, and it came out as 36 nanomolar, so basically confirmed the high potency of the compound. We then set about, we did do a survey of the commercially available compounds around that core, and that was part of Jimmy's thesis work last year. Many of those aminophenylpyrimidines had no attached aryl ring, but did have a lot of variability in the sort of north part of the molecule, so where the amine is attached to the pyrimidine ring. Many of those compounds, about all of them, were basically low activity. A couple which had an aryl ring on the thiophene portion of the heterocycle had some low activity, which seemed to imply we needed an aromatic ring in that position. We then set about doing some synthesis around this core and went for the following substitutions. So there were the original amine on this compound was substituted for a morpholine ring, and then a more complex side chain based on a pyrazine, which was deduced from another active in the Trescantos set. So the compound TCMDC 132385 had the similar core without the sulfonamide, with a sort of a long chain in place of the amine, with a fluorinated aromatic ring attached. So the 3 variations in that position, there was an initial amine, there was a morpholine, and then there was this other side chain. On the other side of the compound, we varied that according to which boronic acids we could lay our hands on and buy. And so several different aromatic rings were trialed, many of which had para substitution rather than meta. Now, in essentially all cases, so all changes, change to the morpholine, change from the meta-substituted aryl sulfonamide, for example, to the para-substituted. Essentially, all these changes completely eliminated activity. In a couple of cases, these compounds, which were all had their C-log Ps evaluated before the compounds were made, and we were very careful to only make compounds which were predicted to be quite soluble, or at least have low log P values. In some cases, there was no opaqueness in the sample, and in some cases there were minor floating specks. But in many cases there weren't, so it didn't affect all compounds by any means. But some of the solubilities were surprisingly low, experimentally, I guess. But the upshot is that this molecule seemed to be extremely tight SAR, and it seemed to be few patterns to the data that we could tease out. So we kind of come back to, at the moment where we stand, is that we kind of come back to the hit compound, which has an amine, that has an aryl sulfonamide, and a pyrimidine ring, aminophenylpyrimidine ring. And we think about whether or not there are simple substitutions that we've overlooked for this series, which we ought to evaluate in order to make sure that we've covered our ground reasonably well. Again, we're looking for low log P compounds, and we're looking for diversity in the synthesis, not necessarily incremental changes, but diversity in the synthesis. Murray and Alice, you've both obviously looked at these data quite a lot. Did you want to say anything about your thoughts about what the data mean, which I might not have covered in my summary there?
I think the only thing that we've discussed, which looks like it's the high potential, is that we're looking at all these compounds and grouping them as one, but I think there's a high possibility there's two modes of actions here. I think the molecules that extend from the amine and extend up, so what's the number? So 137, which was so that without the sulfonamide has a very similar activity. So it's suggesting the sulfonamide is not doing anything, but whereas in 106 compared to, I can't think of the number, basically 106 without the sulfonamide, there's a huge change in activity.
1, 2, 3.
Yes.
So you remove the sulfonamide and it turns to 11.
So yeah, 1, 2, 3 versus 106. There's a huge change. So I think it's not a ridiculous suggestion to say that there's potentially two modes of actions of this with this small core. The core might not be doing anything; it could just be a spacer unit. So I think we can investigate on the amine, or investigate trying to extend the sulfonamide a little, but it's two different areas, because I think modifying both those areas at the same time is not going to give us any activity, or I wouldn't predict it anyway, not at the moment.
Yes, that's right. It is that was a possibility. So yes, 106 is the hit compound. Removal of the sulfonamide, so replacement of sulfonamide with an H, is compound OSMS 123, and that has 0 activity. If you then go back to the hit compound 106, and you maintain the sulfonamide, but you change the amine for the long side chain, the fluorinated side chain, the activity goes down a little bit, a fair amount, but it's an active compound, so it's 1700 nanomolar. And yet, for that compound, when you remove the sulfonamide, the activity actually increases slightly. So that's a compound that we haven't made yet. That is TCMDC 132385. So that's from the original Trescantos library. It has an unfortunate log P, but it's a compound which is listed there in the original set. So there was some possibility that, yes, perhaps we're looking at two different modes of action here, possibly. There was then some suggestion about whether we wanted to validate that finding in the Trescantos library. So this compound with the fluorinated aromatic side chain without the sulfonamide, TCMDC 132385. That's on one of the figures that you guys sent around. Whether we should resynthesize that compound to check for its activity. We had a discussion about whether that's synthetically doable, and I think the consensus is that it was. Fairly straightforwardly.
And I just
without too much effort.
I just lost it at the last purification step. I don't know what went on. So again, I've just ran out of time, but it can easily be remade. In fact, I've started it.
So it's a possible. So Paul, I don't know if you so your gut feeling to these data, as we said, it's extremely tight SAR. What's your sense about? What's the immediate question that we should be asking?
I mean, to me, I think there's two phases. The first thing I'd like us to, or suggest that we consider doing is as a high profile is profiling 106 a little bit more. So have we actually got a measured solubility on that microsomal stability? Profile that through some of the life stages, so we really understand what the strengths and weaknesses of our lead are. So I think that's important to do to put the whole series into context.
Yeah.
So hopefully Sue Sharman can help with the PhysChem and the in vitro DMPK.
We have plenty of compound now.
Yeah, so that would be great to do that. I guess then when you look at the overall SAR, it does look potentially quite a tight SAR, but you could argue that quite a few of the changes you've made to date are fairly speculative and have been driven more by synthetic accessibility. So, some of them, replacing the amine with an aromatic ring, there's no reason why you would expect that to be active. So
yeah, that was a synthetic accident.
Exactly, yes. So you can take that as a strength and a weakness in terms of, it's disappointing we haven't got activity, but it does point to there being specific interactions. And I think really the key now is, does this series have any scope for further optimization, or is it just going to be a singleton or such a tight SCR that we can't move it on? So I would be tempted to have a range of targets at least as a priority, which vary substituents on both the sulfonamide and then the NH2, perhaps taking each to secondary and tertiary, but perhaps acyclic, because we look at the morpholin at the top doesn't look well tolerated, and then to actually build out some things like some aminoethyl or hydroxyethyl to actually get some solubilizing groups on to see if we can pick up another interaction, boost the solubility. So I would make changes both at the sulfonamide and at the NH2, and then I'd also be interested in looking at perhaps putting either one or two ortho substituents onto the phenyl ring, just to see if we can force a twist onto the molecule to hopefully increase the solubility, because the inherent log P is low. So you might expect good solubility, but I suspect it's just because the molecule is fairly planar that it just stacks really well in the crystal state and is poorly soluble. So often just an ortho substituent or two onto that phenyl just to try and throw that aromatic ring out of planarity. If that is tolerated for potency, that could give a solubility boost and take us into a better PhysChem space.
That's interesting. Yeah, we were discussing here. I think Alice suggested that we look for meta di-substituted boronic acids. So the idea would be that one of the substituents would have some kind of interaction, and the other one could be used for enhancing solubility. But actually, it's an interesting point you make that perhaps it is the molecules just too flat and stacking too nicely. The ortho idea is really nice. That would change some of the searching we've been doing for commercially available boronic acids, or things that we can easily synthesize from what we have to hand. But that's certainly a search that we can easily do. Yeah. So changing both at the same time. So yes, I mean the changes which I guess would make sense, as you said, is going to secondary sulfonamides, I guess, things like that. For the amine variation, I think you said hydroxyethyl. You were talking about that position.
As well, yeah. I mean, I would do it at both positions. I would try and sort of build an interactive group off the sulfonamide as well. So to perhaps have a, hydroxyethyl onto your sulfonamide, or you could look at acid and amine just to see if we can pick up another interaction, but using a more polar group, so you're not pushing the log P. And then I would probe the same off of the NH2, perhaps on an acyclic, and then perhaps have one where we just cap one of the NHs off as an N-methyl as well, just to get hydrogen bond donor out again to see if that improves some of the permeability. So
sure.
well, we could go more speculative and look at more diverse changes, but I think it's important to know whether in this actual lead is there scope for varying the two substituents we've got, because if there isn't, and then you're into the game where you've actually got to make very diverse changes to get activity, that's looking like a tough series, and I might be more mindful to then park this series and pick up another one, because then, you know, there's nothing to drive the selection of new series. You're just looking at core changes.
Sure, yeah. Yeah, so I mean, it seems like if we went for, I mean, a reasonably focused second round of 10 or so planned compounds, as well as a couple of others if the chemistry is straightforward. If we went for another round of 10 or so compounds with some of those changes in, if we then saw the same effect, so we saw a series with lots of inactive compounds, then that's looking pretty bad for the series. And so at that point, you think, well, okay, it's just going to be too difficult. But at the moment, there do seem to be some pretty simple changes we could make to these compounds to explore some of those changes. I mean, it seems like I wouldn't want to invest a lot of time in synthetic chemistry. So if we did have any more speculative analogs, it would be because we could get our hands on the starting material. So we're going to do some fairly obvious changes, I think, for the next round if we do that.
No, it makes good sense. And I can see some of these could be synthetically tough. So, you know, getting some of the key boronic acids may be important. I don't know if there's anything we can do. I guess you've done the searches. Are any of them commercially available but harder to get? Is there anything we can do to reach out to companies that might have those and either get them to donate 100 mgs or two, or find ways to
sure. Absolutely, yeah. So for the ortho, that's interesting. We haven't done that search, and that could be something we could think about. We do have a very good supplier. Yeah, Alice. Oh, we've done the ortho. Okay, well, we'll redo that. So yeah, we do have a very good supplier of boronic acids in Australia, who we've been using very productively, and we can approach them if we think that a key boronic acid is required and perhaps isn't listed or is listed as being expensive. Yeah, okay. So Alice has said, well, we do the author search and put it online to see what we can get our hands on. Yeah. So it's sorry, carry on.
I was just going to respond. I see Alice has posted a comment. We shouldn't do any changes on the core. I guess just my thinking on that, I wouldn't do that, because it's thinking, where does that take you? If you change and find a different core, you probably still need to go back and re-optimize, so it then still comes back to, can you find substituents on that core that allow you to further improve the property? So unless you break into a new series, you've still got to address that issue of the substituents. So I'd be tempted to do that first, because I'm struggling to see how just changing the core really moves you forwards where you need to be. But I'm happy to discuss it. I may well have missed something if people have got a strong rationale for why core changes will get us where we need to be.
So yes, we were talking about whether it's possible to access simple changes in the heterocycle by doing the same kind of standard heterocyclic, cyclizations to get, for example, furan rather than thiophene, that kind of change. If that chemistry was straightforward from things that we had to hand, and then if the chemistry to functionalize that was similar, which it possibly could be, then it became something that we were interested in having a go at. But we are in the middle of doing a lot of searches around which other cores might be available. So for example, we searched on changing to pyridines, changing to the furan equivalent furan core, the kind of indole, these other kind of cores which are similar. And it looks for the moment as though many of these compounds, these kind of structures, are coming up blanks. Now, on the one hand, that's great from a chemist's perspective. You think, fantastic, we're in a new area of heterocyclic chemistry. We can get a lot of interesting novel compounds out. But from this project's perspective, it's not so great, because you think, well, how much work are we going to have to put into to make these things? So we're going to finish those searches off to make sure we know where we stand. If there is a compound which we could make, for example, the furan, just for the sake of it, if we can make that with things we have to hand, and we think based on any literature we see that the chemistry is going to be equally easy to the thiena case, then we might have a pop at it. But again, it is more speculative. So, for example, in the I don't know what the ring system is, but the equivalent indole case, where instead of a thiopene, you've got a pyrrole. You're changing from an H bond acceptor to a donor, and I don't know. That's presumably going to have quite a big effect, and it's only worth doing if we can get that compound very straightforwardly. So sorry, I should clarify. The searches we're doing at the moment are for synthetic accessibility, so who's made these compounds before, but also any possible sources of compounds that might have the same kind of core there. We're doing the searches that we do by default now of can we make it? If we can't make it, has anyone made it before? Can we approach those people for possible samples? At the moment, nothing much is coming up, it must be said. A lot of patents and other things. So we would factor that in terms of synthetic accessibility again. I don't think it makes sense to, obviously it doesn't make sense to have all 10 compounds for the next round with changes in the heterocyclic core. There's no point in doing that. It would just be one of the possible variations if it was straightforward.
Sure, and I think what would be good to do as well then, if you have changes to the heterocyclic core, then to go back into, say, the malaria site on Campbell and just research whether they're already hit within the 20,000 that encompass that change, because there may be information we can glean there. So if there's similar groups that are already inactive, so that might help you drive that selection.
Yeah, that's part of the search actually. So in each case, it's a sort of commercial search. It's a SciFinder search, and it's a Campbell search. And pretty much in all the cases we've been searching for the moment, the Campbell's coming up with nothing.
Okay, that's great.
Yeah. Okay, well, so look, as a result of that discussion, I think we need to propose another top 10 as the next thing to do. So we need to come up with a list of top 10 and then alert the community to that, to ask for any changes or feedback in terms of what we should make next. So our action item after this meeting is to then come up with that list of possible suggestions. So we'll try and do that this week, I guess, in the next couple of days, and try and post something to get that discussion started. We have other things to be doing in the meantime. But we can get that list active and then see what people think.
No, I think that would be great. So I could send the suggestions in, and then it might be worth just trying to think of a way of widely publicising that list, so we tap into as wide a set as possible, just to get an online debate and agreement going. So we could set a deadline for that. That might help as well if we said, we posted our suggested 10 in the next week, and then perhaps give a week for people to feedback comments, and then we would then review those comments perhaps and agree that 2 weeks' time we'll lock a list of 10, and then seek people who can help support those syntheses.
Yes, excellent. Okay. Yes, I had an extremely productive conversation with the Royal Society of Chemistry in the last week or so, and I will see if there's interest there in distributing our proposed top 10 list to some of the contacts that they may have, to try and access some people that we perhaps don't access via these meetings.
No, sounds great.
All right. Yeah, okay, so that's actually number 4 is construction of top 10. So we've just covered that, I think, quite quickly. So that's good that we've got that plan in place. So is there any other business that we need to discuss or forgot to discuss, which people would like to bring up?
I think, Matt, just related to profiling the series, it would be good. I can liaise with someone, I don't know if it'll be Alice or Murray in your group, to try and arrange for 106 to be tested, say, for liver stage activity, gametocyte activity, again, just to get some greater life cycle profiling of the compound. So who would be the key contact there?
So I'm away from tomorrow for a week. Alice, in my absence, will be the person to contact about that. I need to contact Sue which I just need to write an email to see if we can get solubility and basic metabolic assays done. But for the kind of assays you're talking about, yes, it would be tremendous if you were able to arrange for something like that. Yes.
No, that's fine. I'll drop you an email then, Alice, and we can sort that through. I can send you the contact details. And I guess the other action probably is it would be good to start to have a mitigation plan. So there is a chance that this series then isn't going to have the SER. So perhaps starting to identify alternate series would be a good thing to think about. So that's certainly
yes, we started that. I think Alice is about to share a link of a wiki page. So Alice already did some analysis of one possible other series from the GSK paper, and just in terms of scoping out what's known, and we sent that back and forward internally, just to discuss what was needed for that that information. And so this is one of the evidences of the productivity enhancements affected by GitHub. I stuck that on GitHub over the weekend, and Alice has already taken care of it. So Alice then put that information on a new wiki page to keep as a summary of other possible series that we're considering, and she's just put the link there. Now that's only one series that we looked at, and it's something that I think we'd like to do for some other series from that key GSK paper. Now, of course, one of the things we don't want to do is duplicate stuff that's going on elsewhere. So it could be that one of the things to do is to clear, perhaps with GSK, if we can, which of those series they think are not being addressed by other people or which are laying fallow, so that we can analyze the right series, right? So that we can spend some time analyzing productive series rather than things that other people are doing already. I don't know if that's a quite general conversation. I don't know if you're happy to have that conversation with GSK Paul at this stage.
So I can take that as an action for sure, Matt. So because I think at MMV we're fairly well aware of quite a few groups. So obviously there's work that's done by GSK, but there's obviously other groups who've also analysed the HTS output and either considered or done some initial work on series. So I'm very happy to check that within our network. So I'll check that series that Alice has thought of already, and I just encourage you, drop me an email as soon as you start thinking of a series, and I can check that fairly quickly just to avoid, as you say, that duplication that people put large amounts of work into searching or thinking about series that may have already been considered or even worked by other groups.
Would you be able to start that process with the series that Alice has got there? I think it's series 1 in that paper. I think is the first one.
Absolutely.
And she's done the search already.
I'll do that this week.
Great, fantastic. Thanks so much. That's good because there's plenty of other stuff to be going on there, but of course on the presumption that no one else is looking at them, which is obviously not right. Okay, but yes, you're right. It's important to have a backup there. Oh, Alice has asked the question: Is it better if we let you know which series we're interested in before we post that information, or which way would you like us to do that? Because we can just do any search.
No, I mean posting it's fine. Perhaps Alice just send me a link when you post the series. It was more just to avoid that people have spent a week, searching the literature and thinking about SER in the series. So I'm relaxed about how you do it, but it's just to avoid you putting work into a series that might have already been considered and dismissed for another reason.
Okay, good, great. Okay, anybody else, any other business, Paul, that we need to discuss now?
No, I think that's been a good focus meeting.
So the general plan, of course, is to have another meeting in 2 months or so. May, June, July, so sometime mid-July, I guess, would be the plan. Depends on how the chemistry goes, depends on what we get back from the biology. Of course, we'll continue to post everything every day openly so people can see where we are, but we would plan to have another one of these in maybe a couple of months. Paul, you and I can talk off this site again just to make sure everything's going okay in about a month or so, but other than that, we plan on having another one of these things in July sometime.
No, sounds good.
All right, fantastic. Thank you, Peter Murray Russ, for those words of encouragement. Okay, that's good. So just remains me to thank everyone for coming. Thanks for the inputs. Of course, the dialogue carries on, mainly on the G+ pages, which are being pretty active at the moment. Also, if any of you are familiar with GitHub and enjoy navigating around, the link was posted earlier. Please feel free to navigate all the issues that are currently open. We do try and label them with what's required. There are some action items there or unresolved issues there which are labeled as search or data needed, which are labeled as that because there's no experimental chemistry required. There's no experimental science required. It's just data analysis and thought about data. We're trying to label things correctly so that people can see exactly what's needed. I'd encourage you all to try that out and see where we're up to. Other than that, I think we can close the meeting. It's about 5 o'clock. So I'll say thanks to everyone for turning up and see you again next time.
Great, thanks for meeting. Have good days for the rest of you.
Yeah, thanks, Paul. We'll see you again.
Yeah, goodbye.